You can start, ma'am. Hello.
Yes. Can I start now?
Yes, ma'am. Yes.
Am I audible now?
Yes, ma'am. Yes.
Thank you. Can we start now?
Aarti ma'am is not there. We'll
Okay.
wait.
Okay.
Hello, Aarti ma'am.
Hi, yes, sir. Yes, yes.
Yes, ma'am.
I'm
You are
sure.
disconnected.
Yes, sir.
You can share the PPT, ma'am.
Yes, sir. Now is it visible, sir?
Yes, ma'am. Yes.
Okay.
Can I start now?
Yes, ma'am.
Yes, ma'am. You can start.
A very warm good afternoon to everyone. This is Debashri Bodhakur, working as an assistant professor. I would like to welcome you all in this lecture series. Today we have with us Dr. R.T. Prabhakar Kamble, working as a lecturer in Osmanabad SAM Center. Ma'am has qualified CSR UGC NET with All India Rank 56. Apart from this, ma'am has numerous publications in reputed national and international journals. Ma'am is also a member secretary of Award Committee of Microbiology Society of India. Now, I would like to request ma'am to take over the session. Over to you, ma'am.
Thank you, ma'am. Thank you very much. Should I start?
Yes. Yes, ma'am.
Okay. So, very good afternoon to all of you. I am Dr. Aarti. Ma'am, I am working in Department of Sadhguru Gadge Maharaj College, Karachi, not in Osmanabad. Okay. So my topic is studies on Actinomycete from soil under cultivation of Curcuma longa. This study is carried out under the guidance of Professor Suhas Kulkarni sir. First of all, I would like to thank Dr. A.M. Deshmukh who give an opportunity to deliver in lecture in a PhD webinar. So, should we start? First of all, we will take introduction of this topic. So, as we know that the soil is a natural and excellent medium, which is useful for most of the organism, and it is useful for their growth and development. From the microbiologist view of the point, it is the most dynamic site of the biological interaction in nature. There are many references out there for that rhizospheric soil, which is influenced by root exudate, which is a selective habitat for most of the organism. For most of the microorganisms, they are live there. Okay, so my topic is we are collected soil samples for the isolation of different varieties of the Actinomycete, and these soil samples are collected from soil under the cultivation of the Curcuma longa plant, which is a turmeric plant, which is known as a golden spice, and which is also traditional crop, and it has been used traditionally in Ayurveda, and it is one of the most important plant and major cash crop in India. Okay, so why the antimicrobial substances which are released from the Curcuma longa, they are diffuses into the surrounding medium of this plant, and so the microorganisms which grow in that area, they become resistant to these plant substances, and so the Actinomycete are one of the major group which are also resistant as antimicrobial substances. So we will, most of we know that what are the Actinomycete. As we know that the Actinomycete, when first rain come from, the beautiful smell is coming from soil, and that is due to presence of Actinomycete produces a geosmin naming compound. What are the Actinomycete we will study? There are some silent features here are described here. That is Actinomycete. It is a connecting link between fungus and bacteria. Though they are bacteria, but they show some character like a fungus. They are being gram-positive and filamentous growth like fungus, and they are well known for the producers of the antibiotic. And they are most of their aerobic, and also they are anaerobic, but they are also widespread in nature with a high GC content. Their GC content is between variable 60 to 70%, and they are important source of antibiotic, not only source of antibiotic, but they also produces plant growth promoting agents. So here are again a few features, silent features are there. They are non-motile, non-capsulated, some are aerobic, and some are anaerobic. And they also produces different varieties of a bioactive agent like vitamin, antibiotic, enzymes, and many growth promoting agents. And they are important microbes in a soil community, and also they are very important for the agriculture application, especially for biological control of insect pests, and also they are helpful for a plant growth promoting option. Here we can see different applications of Actinomycete in medical application. We will also well known for the production of antibiotic, antifungal agent, and they are also producing different MT, MDR, and nature treatment. And in industrial, there are different producers of probiotic. They are producers of plastic bio detergents, and in environmental environmental applications of their, they are producing biofouling, bioremediation. Again, they are also useful for control of radioactive waste treatment, marine bioremediations. They are used, and in agriculture, their applications of Actinomycete, because they are producers of different of the plant growth hormones like siderophore, phosphate solubilization agent, and main bioactive compounds. So these are the varieties of applications in different fields. So there are also few characters here are listed. I have already described this character. So present study, what is our aim of the study? Present study aim at the estimation of the actinomycete under cultivation of Curcuma longa from the rhizospheric soil, which are collected from different villages of around the Barshi, which is a place situated in Solapur district of Maharashtra, India. So I will here describe why the topic is selection. Antibiotic produces different varieties of the secondary metabolites like made antisepticide, antituber, anti-inflammatory, antiviral. These are number of herbicidal compounds. And rhizospheric soil, which are contain abundant of the bacteria, fungi, protozoa, nematode. But in the root exudates of the rhizosphere, which can control the disease, which suppress and help in a nutrient cycle. And the different compounds which are secreted plant root into the rhizospheric, they perform a multiple function. And again, these bacteria play an important role in the rhizospheric soil, and also they have a considerable effect in a plant growth, which protect the root from pathogenic microorganisms. Here are some references for this. In addition to actinomycete, they can produce antibiotic, which reduces the antibacterial activity in a soil. And also, they have play important role in improving medicinal values of the medicinal plant. That is a symbiotic association between medicinal plant and these actinomycete, and they have a role in a nutrient availability, disease resistance, which can aid the quality of the medicinal compound, which is demonstrated in the medicinal plant. And here is a reference for this. And so it is an increasing interest in the research of what is the interaction. We will see what is the interaction between medicinal plant and their rhizospheric microbe for the improvement of the medicinal plant and production of the secondary metabolite of the actinomycete. So there are very few we are seeing there are very few studies are going on this subject. So we have started the subject on the actinomycete between rhizospheric soil is selected rhizospheric soil under the Curcuma longa, because Curcuma longa is one of the most important medicinal plant. So these are the reason for my topic selection, and we are going towards my next, that is the aims and objectives. So we are carried out a chemical analysis of the soil sample. Then we are done the microbiological analysis of the soil sample, which include the study on the glycerol aspergine agar. It is used for the isolation, cultivation, and enumeration of the actinomycete. So we are also enumerated the bacteria, fungus by using nutrient agars and cyprotes agar for fungi. And total actinomycete population we are noted. And we are also carried out the microbiological and cultural characteristics of the actinomycete. And we are done the biochemical activities, enzymatic activities, and sugar utilization pattern of actinomycete, resistance pattern of actinomycete. We are also studying a pesticide resistant pattern of actinomycete and tolerance pattern of actinomycete. And we are carried out the antimicrobial activity against the bacteria, fungi, and yeast. And then we are carried out the 16S RNA sequencing and phylogeny by using phylogeny of only limited, that is the potential actinomycete isolate, which produces broad spectrum antimicrobial substances. And also we are carried out optimization, also we are carried out optimization of the antimicrobial producer substance, which are isolated from actinomycete under cultivation of Curcuma longa. And then we are carried out MIC of this actinomycete. So materials, next topic is a material and methods. In this table, we can see there are 12 different sample collection sites, and there are 20, total 21 soil samples are collected from 12 different regions of the villages, and these villages are situated here. See, we can see these villages are situated in Barshi Taluka, and Barshi Taluka is situated in Solapur district. And in this type of the soil, in this region, we get different varieties of the soil. That is light black, deep black, medium black, and light black, and also red soil. We got. So we are carried out a collection of a soil sample. So soil sample were collected from rhizospheric soil under the cultivation of turmeric plant from 21 different sites of 12 villages, and they are transported to the laboratory in sterile bag. They are air dried at room temperature, and they are used for isolation of actinomycete. So we are also carried out chemical analysis of the soil sample. In chemical analysis of soil sample, we have carried out different chemical parameter, like we have known potassium content, moisture content, pH content, nitrogen content of that soil, and then we have isolated soil actinomycete from these soil samples, and also we have studied the relative population of actinomycete, bacteria, and fungi. And also we are carried out morphological studies. Morphological studies of the actinomycete, it is carried out by using inclined cover strip culture technique, which is devised by Gibbs and Schachter. And then we are also carried out the cultural characters by using colonial characters, different pigmentation production. Pigment production are using shade and different diffusible and non-diffusible pattern. And physiological characters like pH effect, temperature effect, they are also studied. And different biochemical tests are studied different. In the different biochemical tests, we are also studied enzymatic activity, utilization carbon source pattern on Schilling and Gottlieb's media, utilization of nitrogen source, and we are also studying hydrogen sulfide production. And we are identifying the actinomycete on the basis of by using Burgess Manual of Systematic Bacteriology, Volume 4, and also some isolates we are also identified by using microIS software, and some isolates are identified by using 16S RNA technology. And the morphological spore study we are carried out by using light microscope and a scanning electron microscope, which are this study is carried out in physics department of the Shivaji University, Kolhapur. Then we have studied a resistant pattern of actinomycete, a different resistant factor studies of a resistant pattern of actinomycete, and resistance patterns to antibiotic, arsenic compound, and other inhibitory substances. Inhibitory substances in that we have used the arsenic compound, arsenic trioxide, different types of antibiotic like penicillin, streptomycin, gentamicin, and we are also used sodium acide, mercury chloride, and sodium chloride resistance pattern. And also we are studying a tolerance pattern, tolerance for different subject, like we are also studying the different resistance pattern, like a pesticide resistance to the pesticide, and also study different antimicrobial activity of substance by using 7 bacterial pathogen and 9 bacterial pathogen and 7 different fungal pathogen. And then we are carried out 16S RNA sequencing, then phylogenetic trees, and then these are submitted to EMBL, EMBI, and ENI for and we got the accession number, and then we also carried out optimization of antimicrobial study and MIC of that. So the result and discussion in this topic. We are carried out a chemical analysis of 4 different types of a soil sample that we have got: the deep black soil, In the figure, in graph, we can see 4 different varieties of a soil sample and the moisture content. Like deep black soil, it shows high percentage of the moisture content as compared to light black soil. And this is the pH pattern, that deep black soil show highest pH, that is 7.79, as compared to light red. Light red has a low pH. And this is a organic carbon compound. Light red show highest amount of organic compound. And this is a phosphorus content, that is deep black soil. It contains highest amount of phosphorus content as compared to, then followed by medium black, then light black and light red. This type of soil, they contain as compared to equal amount of phosphorus content. And this is the potassium content of soil. Like deep black content, it is the highest quantity of the potassium content. And there is a little bit difference between these 4 types of soil, that is content of potassium. And this is nitrogen content of a soil sample. That deep black soil, again, it shows highest content of nitrogen as compared to this medium black and light black. Also light red soil also contain highest compared, followed by deep black soil. Again, this is a CN ratio, carbon and nitrogen ratio. It is the highest in a light red soil, and then followed by deep black, then followed by light black, and medium black soil contain a lowest CN ratio. And this is percentage of the actinomycete. We found a large percentage of actinomycete in light red soil as compared to all 3 types of the soil. When we had studied statistical analysis of this, there is no significant difference in each of the following 4 cases. Hence, there is no statistically significant difference that all calculated parameter like deep black, medium black, light black, and light red soil type. So these are the number, that is the enumeration of the actinomycete population on different nutrient media. We are using a different nutrient media like glycerol aspergine agar, starch casein agar, Bennett's agar, sucrose nitrate agar, glycerol nitrate agar. As compared to all these, we found on the glycerol aspergine agar, the number of actinomycete show highest actinomycete number. So we are further carried out our further studies is carried out on glycerol aspergine agar plate. This media we have used for whole the study. And also there is a when we are data collected, there is a statistic is a value is a t value, table value is again more than t value, and the test is not significant for glycerol aspergine agar, sucrose nitrate agar, and all these media. So there is also no statistical difference, but we found a more number on glycerol aspergine agar. This is the average value of the actinomycete count on different media. This is the distribution pattern of microbial in different soil. In deep black soil, we get more number of the bacteria, as compared to medium black, light black, light red. Again, there is no statistical significant distribution pattern. So these are the we got. But 124 actinomycete were isolated from 21 different soil samples, and these are collected from 13 different villages under the cultivation of Curcuma longa. So this is the identification and generic distribution pattern of actinomycete. That is the 70.97% are the streptomycin, and only 29.03% are other than the streptomycin. These are the actinomycete isolate on glycerol aspergine agar plate. These are the isolates of actinomycete. So see, other than streptomyces, we also got a culture of streptoverticillium. Number of streptoverticillium and nocardia is the highest, then followed by micromonospora, actinomodura, microbiospora. We also got a number of micropolyspora, actinoplans, ampullurilia, dermatophilus, streptosporangium, and thermoactinomycetes. So this is a spore morphology of streptomyces under a light microscope. Here we can see hooked filamentous structure of spores. And these are the scaling electron photographs of streptomyces spores, which this study we are carried out in the physics department of Shivaji University, Kolhapur. So see, in this photoplate, number of 6, we can see the spore structure of the streptoverticillium. In photoplate 7, we can see a spore structure of nocardia. Nocardia, as we see, a fragmented structure of a nocardia. And in photoplate 8, we can see spore morphology of actinomodura. And in photoplate 9, we can see a spore morphology of ampullurilia. This is the sporulation pattern. When there is a single spore is observed, then that culture we called as a micromonospora. In photoplate 10, the structure of micromonospora we will see. And in photoplate 12, there is a spore morphology of microbiospora. And in photoplate 12, we can see the spore morphology of micropolyspora. And in photoplate 30, we clearly observe the spore morphology of streptosporangium. So this is a biochemical studies of actinomycete. We can study a number of biochemical states here are carried out by different enzymes for the enzymatic activity, study of enzymatic activity on different, like amylase, we have carried out catalase test, caseinase test, cellulose test, chitinase test, then again lecithinase test, lipase test, nitrate reduction test, protease test, tyrosinase, urease. For these enzymes production is a very helpful, because as we know, amylase is used in photo industry, food industry, and textile and paper industry. Catalase is used as an antioxidant and dairy industry. Chitinase is used in a plant pathogen, in plant for resistance against the pathogen, and protease is used to protect the plot and anti-cancerous. There are different wide applications of these enzymes. So we have found the actinomycete, which has ability to produce a varieties of enzymes. Like chitinase enzymes are produced more number of the actinomycete isolate, and then followed by amylase. All here we can see a percentage of the actinomycete is listed here, that 79 point actinomycete isolate produces chitinase enzyme, then followed by amylase, and then all here is a data is here. And this is the also biochemical activity like nitrogen reduction. We can see in photoplate number 14 and TSI, that is the triple sugar ion activity of actinomycete. Here we can see the production of photoplate amylase, casein, urease, and lecithinase production. This study is carried out. And also we have studied a different color pigment which are produced by different actinomycetes. So here is the data. Out of 46 color pigment which are produced by actinomycete isolate, 24 were found diffusible and 22 were non-diffusible color pigment. And which are color pigment, color diffusible and non-diffusible pigment, like yellow color pigment, we have isolated gray color, red, brown color, and total 2 verticillium species produced only gray non-diffusible color pigment, and total 4 species of Actinomycete nocardia produces 2 yellow and 1 brown diffusible, 1 red non-diffusible color pigment. So again, it is a carbon source utilization pattern of actinomycete. Carbon source utilization pattern is done on by using Gottlieb and Shirley media. Like see, here is a number of carbon sources like arabinose, dextrose, fructose, galactose, glucose, insulin, lactose, maltose, mannitol, arabinose, sucrose, xylose, sodium acetate, and sodium citrate. So these are data of actinomycete percentage, which are able to use these carbon sources. This is a generic distribution of actinomycete on the basis of H2S production. So H2S production we have also carried out that out of 124 isolate, 88 were streptomyces and 10 are streptoverticidium, 10 actinomycete isolate from nocardia, 5 micromonospora, 2 species of each Actinomycete microbiospora and micropolyspora, then one species of Actinoplanes, Ampullaria, Dermatophilus streptosporangia, and thermoactinomycetes. So this is a generic distribution of actinomycete which are resistant to various inhibitory substances. Like various inhibitory substances, they are used like arsenic trioxide, crystal white, sodium azide, 2% we are used 0.01 and 0.02% weight by volume, phenol, Dettol, HgCl2. And we see most of the actinomycete, they were resistant to the crystal violet, and then followed by sodium azide 0.01%, and then followed by arsenic trioxide, and then followed by sodium azide 0.02%, then followed by HgCl2 and phenol and Dettol. This is the effect of salt concentration. Different salt concentration we are used. Most percent of the actinomycete, they have tolerate 4% salt concentration, and 67.74 actinomycete, they can tolerate 7% salt, and then followed by 10% actinomycete are resistant. They show a tolerance pattern at 10% sodium chloride, and only few actinomycete isolate, they are tolerate to 30% actinomycete. They are like a halophilic organism. Actinomycetes are there. So here we can see a sodium chloride tolerance pattern of the species which are having enzymatic studies. Streptomyces from this graph, from this table, we can see a culture of Streptomyces exploitis, which can tolerate a 30% salt concentration, and they are able to produce enzyme, a different enzyme, like it produces amylase, casinase, cellulase, chitinase, lecithinase, and lipase, protease, and urease. And also a Streptomyces fulvicinus, it also tolerate a 13% salt concentration, and thermoactinomycete. These 3 actinomycete varieties, actinomycete species, they can tolerate and able to produce different type of enzymes. So they are also very useful actinomycete isolates. And these are the antibiotic resistance pattern. Most of the actinomycete, like percentage, 88.7% of the actinomycete isolate, they are able to resistant to penicillin, and then followed by 83.06% actinomycete isolate. They are show resistant activity against chloramphenicol, and then followed by streptomycin and tetracycline. So in this photo plate, we can see the actinomycete isolate which is resistant to antibiotic resistance actinomycete PSC. And this is a pesticide resistance pattern of actinomycete. In this figure, we can show a different. We have used a different varieties of the pesticide like carbaryl, chlorpyrifos, confidor, diethane, polydol, and quinalofos. Okay, so there are the number of actinomycete like confidor and polydol. For these pesticide, same number of the actinomycete isolate show a resistance pattern, and then followed by chlorpyrifos, then carbaryl, and then quinalofos. So this is the again generic distribution of the actinomycete on the basis of temperature and pH. So see, at room temperature, most of the actinomycete show growth, and then followed by 37, and then followed by 45 degrees centigrade. No actinomycete isolate will be found which show growth at 10 degrees centigrade. We found one actinomycete isolate which show activity, which show growth at 55 degrees centigrade, and that is thermoactinomycetes. And as we know that actinomycetes are bacteria, so most of the actinomycete grow at a neutral pH, that is the 7 pH, and some actinomycete isolate like 91.12 actinomycete isolate show activity at 9 pH, and then followed by pH 5. So this is antimicrobial activity of actinomycete. So 9 bacterial pathogen we have used to study the antibacterial activity of actinomycete. Antibacterial activity, we have studied primary screening method, and then agar overlay technique is used for that. So these are the Bacillus megaterium, the culture of Bacillus megaterium, Bacillus subtilis. We can see the activity against Escherichia coli, Klebsiella pneumoniae, Proteus vulgaris, Pseudomonas aeruginosa, Staphylococcus aureus, Salmonella typhi, and Xanthomonas auxinopolis. Most of the actinomycete show antibacterial activity against Staphylococcus aureus, and then followed by Bacillus subtilis, and then Bacillus megaterium, then followed by Escherichia coli, then Pseudomonas aeruginosa, and then followed by Xanthomonas auxinopolis and Salmonella typhi. So this is primary screening of antibiotic producing, and that is Streptomyces flaviviridis AK2. This is the in middle side we have isolate we have strict antibiotic producer organism, and at the perpendicular there are pathogens. Pathogens are strict 1 centimeter apart from that antibacterial actinomycete producing organism. And then here we can see the bacterial pathogen. They are growth up. They are very when their resistance, they are grow along the actinomycete culture, and when they are sensitive, they grow apart from antibiotic producing organism. In this picture we can see this. So this is the antibacterial activity of actinomycete against Xanthomonas auxinopolis and against Staphylococcus aureus. So here we can see antifungal activity of actinomycete, a different fungal pathogen we have used, the Aspergillus niger, Mucor, Penicillium chrysogenum, Rhizopyrus, Trichoderma, Candida albicans, and Cerevisiae. So most activity we have antifungal activity found against Candida albicans, then followed by aureus, then against also Mucor we found antifungal activity against Penicillium chrysogenum, Aspergillus niger, So this is antifungal activity of Actinomycete against Trichoderma species and against Penicillium species. So this table shows a genetic distribution of Actinomycete, which is antagonist to gram-positive bacteria, gram-negative bacteria, and fungi. Out of 88 streptomyces, we found that 42 Actinomycete isolate show antibacterial activity against gram-positive bacteria, and 39 Actinomycete show antibacterial against the gram-negative bacteria, and 30 Actinomycete isolate show antifungal. And then out of 10 streptoverticillium isolate, 4 show antibacterial activity against gram-positive bacteria, and 2 isolates show activity against gram-negative bacteria, and 4 isolates show antifungal activity against the fungi. Like this, we will see the table summarize all the antagonistic activity of gram-positive and gram-negative bacteria and against the fungus. So this is antagonistic activity of Actinomycete against different soil. The Actinomycete isolate, which are isolated from light black soil, they show most of the antibacterial activity against gram-positive, then followed by gram-negative activity, and then followed by fungi. And in the medium black soil, the Actinomycete isolate show most of the Actinomycete show antibacterial against. This is the they show most activity against the fungi. And this is EMBL accession number of the Actinomycete isolate producing broad spectrum antimicrobial substances and their phylogenetic identification. The culture number 15.2 and culture number 18.1. We got the accession number, and this is the identification of the isolate. That is the streptomyces AK1, partial 16S RNA sequencing gene, and strain is AK1. And the culture number 81, we got accession number for that ELK392395, and it is a streptomyces flaviridis, which is partial 16S RNA sequencing gene and strain AK2. That is EMBL. It is a European Molecular Biology Lab. We have submitted the culture. 16S RNA sequencing we have submitted there, and we got this accession number. And in this diagram, we can see it is a phylogenetic tree and 16S RNA sequencing of Actinomycete, which produces a broad spectrum antimicrobial activity. And the name of that culture is a Streptomyces species AK1. We have selected the name AK1, A for RP, and K for my guide Kulkarni sir, Dr. Kulkarni sir, professor. This is the 16S RNA sequencing of Actinomycete broads producing antimicrobial, which are submitted to EMBL, and we got obtained. This is the accession number of this culture. And these are the forward and reverse primer which are used for sequencing. This is again a phylogenetic tree of 16S RNA sequencing of a second culture of Streptococcus, which show a broad spectrum antimicrobial activity. That is the Streptomyces flaviridis. And these are the primer, forward primer, reverse primers, which are used for that. And then we are carried out optimization of a fermentation, which produces antimicrobial substance by using the culture Streptomyces species AK1, because it shows highest antimicrobial activity against as compared to AK2. So the media which we are selected, we are carried out the study, and from the study we are carried it on the media, that is a nutrient glycerol asparagine agar. It show highest activity as compared to peptone yeast agar, sucrose nitrate agar, starch casein agar, soybean agar. And by using, these are the use of different carbon source pattern. Again, we show most highest antimicrobial activity on the glycerol carbon source as compared to all carbon source like arabinose, mannitol, glucose, xylose, maltose, and sodium acetate. And these are the nitrogen pattern, nitrogen source. So KNO3, it shows the highest activity, highest antimicrobial production activity when we use the KNO3 as a nitrogen source. And at the percentage of 3% when sodium salt concentration, it shows maximum antimicrobial activity. And again, the pH is 7. It shows highest antimicrobial activity at pH 7. And at 37 degrees, we found that at 37 degrees centigrade, it shows highest activity as compared to room temperature. And this is the antagonistic activity of the antimicrobial substance produced by Streptomyces AK1 by using glycerol asparagine broth. And so we are a newly devised media by using all combinations, all parameters we are combined and then compiling by all collaborators, and then we devised a new media. And when we are show the antagonistic pattern compare, then it show highest activity. The highest activity is showed by Pseudomonas aeruginosa, then Staphylococcus aureus, then followed by Staphylococcus aureus, then followed by mucor. This is a MIC study in vitro concentration of study of antimicrobial extract, which are isolated from Streptomyces AK1. Test organism Staphylococcus aureus, it show 1:1%. The MIC is calculated in volume by volume percentage. Sodium aeruginosa, it show 0.66 as to 1. As we know that when there is a list, antimicrobial substance is required to stop the growth of substance, to growth of our microorganism, means it is a more effective. So again, Pseudomonas aeruginosa, it is the most effective as compared to fungus, because mucor, it required 4%, 4:1. And again, Saccharomyces cerevisiae, it required 2.33 as to 1. So this is a well diffusion method study of MIC, zone of inhibition, MIC study of Pseudomonas aeruginosa and Staphylococcus aureus by using Muller-Hinton agar plate. So this is the summary and conclusion. When we summarize our study, that most of the actinomycete, they are present in deep black soil as compared to medium black, light black, and light red type. 124 out of total 124 actinomycete isolate, 84 were identified at up to specific level, and remaining 40 were identified up to generic level. And all the cultures are placed in the genera Streptomyces, Streptoverticillium, Nocardia, Micromonospora, Microbispora, Micropolyspora, Actinoplanes, Ampiluria, Dermatophilus, Streptosporangium, Thermoactinomyces. And that is 47 were 47 out of actinomycete isolate, 47 were obtained from deep black, 35 from medium black, and 31 from light black side. And all the genera, they show biochemical active. They are biochemical active and produce different types of the enzymes, and they utilize carbon source and nitrogen source. And most of the actinomycete genera, they are resistant to different inhibitory substances like crystal violet, phenol, Dettol, sodium mosaic 0.01%, sodium mosaic 0.2%, volume by volume, and mercury chloride, sodium chloride, antibiotic, and pesticide. All the genera of the actinomycete were resistant to all tested pesticide except quinalophos. And we found that most of the actinomycete genera show growth at a room temperature 37 degrees centigrade 45 degrees centigrade, except actinoplans was not showed growth at 45 degrees centigrade. Only thermoactinomycete showed growth at 55 degrees centigrade. Regarding the pH, a total 93.55, 100% and 91% of the actinomycete, they show ability to grow at pH 5, 7, and 9 respectively. And the Streptomyces AK1, as we already studied, that it showed a higher broad spectrum antimicrobial activity as compared to Streptomyces flaviridis. And their optimum parameter of AK1, like a carbon source is glycerol, nitrogen source KNO3, salt concentration 3%, and pH is 7%. And maximum production of antimicrobial substances isolated from the Streptomyces species AK1. And incubation temperature is 37 degrees centigrade and time period is of 5 days. And MIC data that AK1 was over 1% show against gram-positive bacteria Staphylococcus aureus and 0.66 as to 1 against the gram-negative bacteria. And these are the conclusion that actinomycete, which are intermediate to bacterial and fungal population. Streptomyces was the most dominant genus, which is followed by Nocardia and Streptoverticillium. And the actinomycete, they have a role in the decomposition of the complex organic matter and pesticide found in nature and agriculture waste degradation. These inhibitory substances of the actinomycete, they used for abatement of soil and water pollution, which is caused by industrial effluent, and it is eco-friendly. And the utilization pattern of the actinomycete isolate, they can be used carbon source and nitrogen source have more importance in industries for designing of a new nutrient media for actinomycete. And the actinomycete from soil under cultivation of Curcuma was first time reported to be resistant to all studied inhibitory agent, antibiotic, and pesticide. And the Streptomyces AK1, it showed the broad spectrum antimicrobial activity against the known human and plant pathogen. So they can be used to prevent the disease caused by these tested human and plant pathogen. So this is a future scope in industry. This an interesting scope for the further research would be to improve the antimicrobial substance production of Streptomyces AK1 by using genetic engineering, and we will use it against the pathogenic or microorganism to cure the disease. And the actinomycete, we show antifungal activity against the mucor. It will be used to cure the pathogenic disease, mucormycosis, which is observed in post-COVID patient. And the enzymes producing actinomycete, it can be used to the different industry for the enzyme production. Carbon and nitrogen utilizing pattern of the actinomycete will be helpful for designing of media for actinomycete. These are the agriculture's use, that is the Streptomyces exploitis, Streptomyces fulvicinus, thermoactinomycete, which showed 30% tolerance of sodium chloride. They can be used, and enzyme, they also produce enzyme, different enzyme, and it can be used as a biofertilizer and agriculture waste degradation in saline soil. And the actinomycete isolate will show antibacterial activity against Xanthomonas oxynopodis. It will be shown to cure a plant disease to control plant pathogen. Halophiles, that is the salt-tolerant actinomycete, will be useful host for the production of antibiotic, enzyme, and biofuel in bulk with low cost, since the halophilic actinomycete have antimicrobial activity and other extracellular enzyme production capability. So there is a less chance of the contamination during fermentation. And therefore, major cost for the sterilization of cultivation media, it will be reduced. And the isolate will show H2S production, extracellular enzyme like amylase, urease, protease, cellulase, nitrate reducing. They can be used for plant growth promoting attributes. And for the environmental bioremediation, we can use by taking field trial pesticide degrading actinomycete will be used to control soil and water pollution and decreases remediation cost. And there is a wide scope for study of absorption and absorption abilities of the isolated soil actinomycete, and for that further research is necessary. So there are a number of references that are used for the study, but few are listed here. And I am again very thankful to Microbiology Society of India and Dr. A.M. Deshmukh, who give a golden chance to deliver a lecture, and my guide, Dr. S.W. Kulkarni. Thank you.